Okay, this is the video lecture for the second half of Chapter 16 on DNA. You may remember from the first half of this video lecture, or chapter rather, that we had been talking about the history of research into DNA. We talked about Watson and Crick. We talked about Rosalind Franklin and her work with DNA structure using X-ray crystallography, all that sort of thing. In the second half of this chapter, for this video lecture, we're going to talk about how it is that DNA actually copies itself. That's a process called DNA replication. And again, that's what happens during S phase of the cell cycle. We talked about that briefly back in the chapter on mitosis. So basically, this is a biochemical reaction, all right? We're not going to go into it in the detail that we did cell respiration, but there are a number of proteins that come into play here, about a dozen or so actually, at certain stages in this process to make, or to make it possible rather for DNA to be copied. And the fact that DNA has a very specific structure, that double helix that we talked about, is manifest in the function of DNA, right? This relationship we've talked about between structure and function. DNA is put together in such a way that it's able to do its job, which is to basically store hereditary information. But the structure of the molecule also plays a very important role in sort of dictating the way that the molecule has to be replicated. And we'll focus on that quite a bit here in a little bit. All right, so Watson and Crick noted that the specific base pairing, remember A with T, C with G, for example, suggested a possible copying mechanism for the genetic material. Watson and Crick assumed that since the two strands of DNA are complementary, meaning if you have an A on the left-hand side, you must have a T on the right, C on the left, G on the right. Because the two strands are complementary, they assume that each strand acts as a template for building a new strand onto itself during replication. And it turns out that's exactly what happens. We have a template replication mechanism here. Essentially, the two strands of DNA that are connected by the hydrogen bonds between the bases, which you actually see in this figure that we talked about last time, the two strands act as templates. They're split apart from each other during replication, sort of unzipped like a zipper basically. And then if you have a guanine on the left, a cytosine would be added on the right as a new strand is built. If you had a thymine on the right, an adenine is added on the left because of the base pair complementarity rules, A with T, C with G. Now, in DNA replication, the parent molecule unwinds. It can't be copied when it's in the double helix conformation. It has to unwind and lay out flat, like a ladder, basically. And the two new daughter strands are built based on the base pairing rules onto each of the two template strands. So here's the basic idea. Now, this is called Watson and Crick's semi-conservative model of DNA replication. This is the one that we know is correct. Turns out they had come up with two other models, which in hindsight, I'm not really sure how they could have thought of as being possibilities, and I guess maybe they were just covering their bases. This one is much simpler than the other two, so we're not going to go into those. Those aren't correct anyway. So basically what we have is a parent DNA molecule here, shown in dark blue. All right, note the base pair complementarity, adenine on the left, thymine on the right, cytosine here, guanine here, and so on. All right, what's going to happen basically is that an enzyme will come in and cut these hydrogen bonds and separate the two parent strands from each other. Okay, those now can serve as templates. So another enzyme called DNA polymerase that we're going to talk about quite a bit here in a little bit basically moves down each of these two strands and it reads these unpaired bases. When it comes to an adenine, it finds a nucleotide with a thymine base on it and attaches it there. Goes to the next one, it says, okay, this is cytosine. It finds a nucleotide with a guanine floating around in the nucleus and attaches it there. So it's adding complementary nucleotides on each of these two parent strands. So what you end up with are two copies of the original molecule, each now consisting of 1/2 original parent DNA shown in dark blue and 1/2 new or daughter DNA shown in light blue. If you look at these two sequences, G, C, A, T, T, A, and so on, you see they're exactly the same. Compare these molecules to our original, and you'll see they're exactly the same. So this is what I mean by a template copying mechanism. Each of the two original parent strands, once they're separated, serves as templates for building daughter strands onto themselves. Okay? This is a semi-conservative model because what you end up with are two new copies of the molecule, each one semi-conserved in the sense that half of it is original parent DNA, half of it is new daughter DNA. Semi-conserved. All right, so the copying of the DNA molecule is really quite remarkable in terms of its speed and its accuracy. And again, there are about a dozen different enzymes and other proteins that come in during various parts of this process at certain parts of the sequence to make sure certain things happen. Let's talk about the accuracy of DNA replication first. There's an analogy here I always use to sort of help students get their head around this. First of all, any mistake that's made during DNA replication, for example, if you have an A and you should have a T added onto it, but a C is added onto it instead, that mistake is called a mutation, okay? There's a base or a background rate of mutation that's very, very low. So mutations are very rare, all right? But it turns out there are a lot more mutations that occur, but they don't make it into the final DNA copy because there are proofreading mechanisms that the cell uses to go back and fix most of those mutations, most of those errors, okay? But some do slip through. And they form the basis for all the genetic variation that we see around us, all right? Okay, so let's talk about the speed of this process. So imagine you walk into a gymnasium and there are 500 typewriter tables sitting there on the floor of that gymnasium. Each one has a chair behind it, and next to each one, there's a complete 26 volume set of, let's say, Encyclopedia Britannica, right? And so A through Z volumes, okay. A typist comes in and sits down at each one of these tables. So 500 typists come in, and they pick up volume A of the encyclopedia, start at page 1, and they start typing. They start copying the volumes of the encyclopedia by typing them out. So 500 people doing this, you end up with 500 copies of that 26 volume set. Imagine how many letters that must represent. How many letters are in those 500 copies of that 26 volume set. All right, at the end of all that typing, one single typist has made a single one-letter typo, and that is a mutation. All the rest of the letter sequences in those 26 volume sets, all 500 of them, are accurate, 100%. But there's one letter out of place after all that typing. That's about how accurate DNA replication is. It's extremely accurate, all right? Okay, and it's also very fast, as we'll see here in a little bit. Okay, so what's going on here? Well, basically, we have to have a place along the DNA strand, or multiple places, where the DNA molecule separates, where the two strands separate from each other. Those points where the two strands separate are called origins of replication. Origins of replication, all right? And basically what you have something sort of like this, okay? So you have double-stranded DNA here, double-stranded DNA here, and here what you have are parent strands that are separated from each other. So that point right there in the center where they separated initially, that's called an origin of replication. And it makes what's called a replication bubble. So inside of this bubble is where all the DNA replication work is going on, because here we have a parent strand and the other parent strand, and they're separated, and so you'll have new DNA, daughter DNA being built onto that, and daughter DNA being built onto that. All right, so all this replication we're getting ready to talk about happens inside that replication bubble, because the center of that bubble was at one point an origin of replication. Now, bacterial DNA molecules, because they're so small compared to eukaryotic, usually have one origin of replication on that circular DNA molecule. But eukaryotic DNA, because it's so much larger, you'll have usually thousands of origins of replication sort of peppered along the length of each chromosome, and each one of those is a place where the two parent strands can separate from each other and copying can begin, okay? And replication is going to proceed in both directions away from that original origin of replication, all right? So that replication bubble that you see there in the middle is going to get bigger. Okay, so here's essentially what we have. So let's look at the left first. This is E. coli. So this is prokaryotic or bacterial DNA replication. We have a single DNA molecule. It's one chromosome. It's a loop of DNA. Usually it's anywhere from 25,000 to 100,000 bases. Contrast that with human DNA, which is about 3.2 billion bases. That's billion with a B, all right? So bacteria have a lot less DNA. They have a single origin of replication, and that's the point where the two parent strands, again shown in darker blue, separate. We have a replication bubble forming here. Notice that we have daughter DNA in lighter blue or gray being built onto the template parent strands like I showed you before. All right, move down to this image. This point here where you can imagine the two parent strands unzipping that way, and over here unzipping that way, that point is called a replication fork, okay? That's where all the action is. That's where all the copying actually takes place, as we'll see. So this replication fork is unzipping that way. This one's unzipping that way. Here you see it further on in progress. And eventually the two forks join up on this side, and we get two separated DNA molecules that should be, with the exception of the occasional and very rare and very random mutation, they should be exact duplicates of each other. Again, notice that they are now 1/2 original parent DNA, 1/2 daughter DNA, semi-conserved. This is an electron microscope view of that very thing. You have a replication fork here moving this way, a replication fork there moving that way along the circular DNA molecule, and here you have the replication bubble. All the copying is happening there and there, okay? This DNA here has yet to be copied. All right, so that's bacteria. Now, let's look in eukaryotes. So again, because eukaryotes have so much more DNA, hundreds of times more DNA in most cases, we have to have thousands of origins of replication, again, as I said, sort of strung along the length of our chromosomes, and each one of those is a place where a replication bubble can form. So again, a replication bubble here, this fork moving that way, this fork moving that way, this fork moving this way. So these two forks are actually moving toward one another. They'll join in the middle. You see that that space in the middle has gotten smaller as the replication bubbles have gotten bigger, right? So eventually what happens is, that all of these replication bubbles will fuse together, and when the last two have fused together, we have two complete daughter DNA molecules, okay? Two completely copied DNA molecules. Again, each one half original parent DNA, half daughter DNA, so semi-conserved. That's what you see in this electron microscope view. Here is one replication bubble with a fork going that way and this fork going that way. A second one here, fork that way, a third one here, this fork moving this way, and this fork moving that way. All right. So in this way, even though eukaryotes have so much more DNA, because the DNA is being copied at thousands of different places along the length of each chromosome, the DNA can still be copied relatively quickly. All right. So now let's talk about what's going on at the replication fork. So again, there are a number of different proteins that are involved here, okay? So we're going to talk about the major proteins here and their primary functions. All right, so the first class of enzymes that are involved here are called helicases. The helicases are enzymes that untwist the double DNA helix at the replication forks. So these are the enzymes that are going to lay the DNA out flat like a ladder because it can't be copied when it's, coiled up like a spiral staircase or in the double helix conformation, as we call it. Helicases are very important. They lay it out flat like the ladder in what's called the ladder conformation. All right. Now, DNA is unstable when it's laid out flat in the ladder conformation, so it wants to coil back up. So what happens is that we then have another group of proteins. These are not enzymes. They're called single-stranded binding proteins. They sort of act like tacks or nails. They come in and they attach to the unwound strands and they sort of lock them down so that they can't coil back up. All right, that's very important. Then we have another enzyme which has a really sort of unique job. It's called topoisomerase. Sort of a mouthful. Topoisomerase. Topoisomerase corrects a problem that occurs between replication forks. So let's say that you have DNA being copied here, right? So we have two replication forks. So we have one here and we have one there, all right? And so this replication fork is moving in that direction and this one is moving in that direction. And again, this is still double-stranded DNA, parent DNA in the middle. All right, we have a replication bubble on the right and one on the left. Well, what happens is that as this replication fork unzips in that direction and this one unzips in that direction, you get overwinding of the parent strand in between and a knot will tend to form. All right, a knot will tend to form. If you think about this, it makes perfect sense. It's exactly what's going to happen because this one's winding in one direction, that one's winding or unwinding in another direction, and you get a knot forming in the middle. And if that knot is not taken care of, the replication forks, when they hit that knot, will not be able to go any further, and the DNA bound up in the knot would not be able to be copied, and that can be a serious problem. So what topoisomerase does is it basically makes cuts. It's an enzyme, and it'll make a cut right here, and it makes a cut right there in those two sugar phosphate backbones. That then allows the section in the middle to swivel and release the tension. All right. Then those breaks in the sugar phosphate backbone are sealed up. The forks continue moving closer. Overwinding continues again. Topoisomerase comes in, makes another cut here, and maybe another cut there. The section in the middle that was overwinding releases the tension and goes back to normal. So that's what topoisomerase does. It prevents overwinding by making those cuts in the sugar phosphate backbone, allowing that section in between that was overwinding to unwind and relieve the tension. All right, and here we can kind of see some of this. Okay, so we have a replication fork right here. Okay, that fork is moving in that direction. Imagine this is a zipper, and it's literally unzipping in that direction. Okay. Here we have our helicase enzyme. This is the one, it's not really shown here, but it's unwinding this DNA here, which is actually bound up as the double helix, like a spiral staircase. It's unwinding it, and it's starting to break the hydrogen bonds between those bases. So it's separating the two strands. Well, again, those two strands want to rejoin and coil back up, so we need single-stranded binding proteins to attach and lock them down so they don't do that until they can be copied. All right, here we have topoisomerase. You can see it's made a cut right there. There would be one further down this way working on the other side because there's a replication fork down here, if you recall, moving in that direction. All right, but you see that cut it made right there in that sugar phosphate backbone. Okay, we also have another enzyme called primase, which I didn't talk about yet. Primase is shown right here. Primase has a really important job. What it does, excuse me, is it lays down what's called a primer, an RNA primer. This is a primer of nucleotides made of RNA rather than DNA. So those are RNA nucleotides, not DNA nucleotides. The purpose of the primer is to create what's called a free 3' end right there. Free 3' end. Let me write that down. A free 3' end. That is very important because the enzyme that actually is going to build the daughter strand, the complementary daughter strand, onto this parent strand, which is called DNA polymerase, we'll see that in a minute, it can't just sit down any old place on that parent strand. It has to have a starting point. It has to have a free 3' end that you see right there, and that's what the primer gives it. The primer gives it a starting place. Primase builds that primer. The primer is usually about 10 or 15 bases in length, but it's just a strand of RNA. Later on, once the primer is no longer needed, that RNA primer, the RNA nucleotides, will be plucked out and replaced with DNA nucleotides. So it's just there temporarily. All right. Okay. Also, one more important thing. Remember, the DNA molecule is anti-parallel. That means that we have two strands that run parallel to each other, but in opposite directions. This is the 3' end on this one, 5 down here. This is the 3' end on this strand, and 5 is down here. They're anti-parallel. This has really important implications for how the two strands are copied, as we're going to see in a little bit. The reason for that is because DNA polymerase, the enzyme that does all the work here, can only move in the 5 to 3 direction. This enzyme, DNA polymerase, can only move in the 5 to 3 direction. So, by starting at a 3' end, it's moving in the 5 to 3 direction by constantly adding on to an existing 3' end, so it's moving 5 to 3. Remember that. That's a really important rule. Okay? All right. So, this primer is very important, like I said, because it gives that enzyme, DNA polymerase, that important 3' end as a starting place. Okay? All right. Now, let's talk about DNA polymerase. So let me write this down. So DNA polymerase is the enzyme that we usually talk about as sort of the workhorse of DNA replication. This is the enzyme that's going to build the daughter strand onto the parent strand. Now, there are actually 3 of these, DNA polymerase 1, 2, and 3. And really, the differences between them are not that important for this level of a course, but there are 3 of them. We'll see a difference between 2 of them here in a little bit. And they do require a primer, like I said. Okay, DNA polymerase works pretty fast. Typically in bacteria, it is going to zip along that parent strand and add nucleotides at a rate of about 500 per second. So it's adding nucleotides at about 500 per second. In eukaryotes, such as ourselves, right, it's a little slower. It moves at about 50 nucleotides per second. Per second. So that's still pretty fast, okay? But the reason why it can move more slowly in eukaryotes is because, again, you have DNA polymerase working at thousands of different locations across that chromosome where each origin of replication was. All right, let's see what's happening here. Okay, this is the basic idea. So we have our parent strand, again, shown in darker blue, our daughter strand shown in lighter blue. On the daughter strand, this is our 5' end, that's the 3' end. On the parent strand, this is 3' end, and that's 5' end. So DNA polymerase, as just shown as this box here, it's actually sitting right here on the parent strand. And on the parent strand, it's moving in the 3 to 5 direction. But that means that it's building the daughter strand in the 5 to 3 direction by constantly adding on to a new 3' end. So it's building 5 to 3. All right. Just like if you're standing in a line and someone comes and stands next to you on your right-hand side, then someone comes and stands next to that person on their right-hand side, the line of people is building to the right. That's all we're talking about here, okay? All right. Now, notice there's no ATP being used here, but this is an anabolic process. We're building a DNA molecule. The reason there's no ATP involved is because each nucleotide, in this case we have a thymine nucleotide, comes preloaded with 3 phosphates. I said back in chapter 8, basically when I was talking about ATP, that it's a modified nucleotide. It's a modified adenine nucleotide, okay? Adenosine triphosphate. It has an adenine, a phosphate, a sugar, and 3 phosphates. So each nucleotide, those carrying thymine, those carrying adenine, cytosine, guanine, comes preloaded with its own fuel tank, basically. So when this comes in, this phosphate will attach to the sugar on the previous nucleotide that was added. These 2 phosphates come away as what's called pyrophosphate, now inorganic. They drop away. And that nucleotide is now attached, as you see, right there. That phosphate that I'm pointing to is this phosphate. All right, see how it's now attached to that sugar right there. Okay, so polymerase adds that one. It then goes to the next, says, okay, there's a C there. What nucleotide does it need to find? One with a guanine on it, because cytosine and guanine bind. And this is all happening inside the nucleus of a eukaryote, right? So there are nucleotides floating around by the billions as a supply inside that nucleus, Polymerase finds one with the G on it and sticks it there. Goes to the next one, whatever that is, finds the complementary nucleotide, sticks it right there. So it's building this daughter strand in the 5 to 3 direction. All right, one at a time, one nucleotide at a time. In eukaryotes, 50 nucleotides per second. In prokaryotes, about 500 per second. Okay, so now we're going to talk about this anti-parallel structure of the DNA molecule and how that affects DNA replication. Remember, DNA polymerase can only add nucleotides to a free 3' end of a growing strand. So a new DNA strand can only elongate or be built in the 5 to 3 direction. You've got to remember that, or you're not going to understand why there's a problem here or the fix for it. Okay, so let's look at what's going on. So up here we have a replication bubble, right? We have a replication fork moving that way and a replication fork moving that way. Okay, we have two strands that are being built. We have what's called the leading strand and what's called the lagging strand. Right now we're going to focus on the leading strand. The leading strand is the easy one to understand, all right? Down here, what you see is what's called leading strand replication. Okay, let's look at our molecule here. So we have our double-stranded DNA, parent DNA, 5' end here, which makes that 3' end. All right, this is our 3' end, which makes that the 5' end. So this top strand is going to be our leading strand, all right? So if this is the 3' end of the parent strand, on what will become the daughter strand being built here and starting off with the primer, that's going to be the 5' end, right? That's the anti-parallel, right? They're backwards, okay? So this end of the primer, before DNA polymerase that you see here sat down, that was an existing 3' end. That's why the primer is built. It gives polymerase, this enzyme, that 3' end as a starting point. And we said that polymerase, by adding to a 3' end, is constantly building in the 5 to 3 direction, all right? So once it starts down that primer, it just goes. It keeps up with the rate at which nucleotides are being separated, the rate at which this fork is moving in that direction. It just sits there and has that parent strand being fed through it, and it's reading these bases in eukaryotes at 50 bases per second and adding the complementary nucleotides to each of these now separated parent bases, all right? That's leading strand replication. You can see how that strand's being elongated here, as if the polymerase is following that fork as the fork moves down, okay? That's called continuous elongation in the 5 to 3 direction. All right. So leading strand replication is pretty straightforward, okay? What I need to do next is do a drawing to show you why lagging strand replication is more complicated, okay? Why there's a problem. So let's imagine that we have a replication bubble here, all right, like we saw in the previous slide. So here we have parent DNA that's yet to be copied, all right. Let's say that this is our 5' end on this side, which makes this end over here 3'. That then makes that 5' and this 3'. Okay, we're going to focus right here on this replication fork, all right. So let's say that we have a strand of DNA being built right here, a daughter strand. Okay. If this is our 3' end, and that up there would be the 5' end, that makes this what? That makes this the 3' end, and that the 5' end on that daughter strand, all right? Okay. Now, what do we have down here? Okay, well, if this is 5', that would be 3'. That makes this 5' and this 3'. Okay, this daughter strand is the leading strand. That's the one I just described in the previous slide. This one is called the lagging strand. This is where the problem is. Okay, so what's going on here, what the problem is that remember that DNA polymerase has to build the daughter strand in the 5 to 3 direction. Okay, well, on the leading strand, The 5 to 3 direction is that way. It's in the same direction that the replication fork is moving. This replication fork is unzipping that way. This daughter strand, the leading strand, is being built in the same direction because there's your 3' end. By tacking onto that 3' end, it's moving in the same direction as the replication fork. Now, based on that, you should recognize the problem on the lagging strand. The lagging strand has to be built in that direction, 5 to 3. So if it's being built in this direction, what's the problem? That's in the opposite direction from the fork. The fork is going this way. This lagging daughter strand is being built in the opposite direction from the fork. Well, why is that a problem? It's a problem because the replication fork is the only thing providing unbound nucleotides, parent nucleotides for DNA polymerase, which is sitting right here to read. So there's a conundrum here. DNA polymerase, which is sitting right here, has to keep up with the replication fork, which is moving that way. But on this side, it has to build the daughter strand in the opposite direction. How does it do both of those at the same time? How does it move in the same direction as the fork, but also build the daughter strand in the completely opposite direction, 180 degrees away? How does that happen? That's the problem here, okay? Now, there's a fix for this, but to understand it, you have to first understand the problem. All right. Now, let's look on the other half of this replication bubble. So let's imagine we draw an imaginary line here, bisecting it in two. On the other half, the fork is moving that way. That means that this is going to be the lagging strand on that side, right? And this will be the leading strand on the other side, right? So on the opposite half of the replication bubble, everything is flipped around the other way. It's the reverse, all right? So you'd have 5' there, 3' here, you'd have 3' there, and 5' there, okay? All right, so hopefully you now understand the problem. And we're going to, again, talk about it either as the problem on this lagging strand or on that lagging strand. These two leading strands, there's no problem, okay? All right, basically the workaround here to this problem is that DNA polymerase, on the two lagging strands, it actually builds that daughter strand in a series of fragments. These fragments are called Okazaki fragments, named after the Japanese scientist who figured this out, all right, Okazaki fragments. And this is the solution that allows DNA polymerase to at once keep moving toward the replication fork, but also to build the daughter strand in the opposite direction away from the fork, all right? These Okazaki fragments, once they're completed, are going to be sealed together into one continuous strand by an enzyme called DNA ligase, all right? I'll show you how that works here in a little bit. Okay, let's look at a figure here. All right, there's a lot going on in this figure, but we're going to take it step by step. So here we have our replication bubble. We have a leading strand there, one primer, right? Polymerase, once it sits down the primer, it just goes and follows that fork because it's moving in the same direction the fork is by building that daughter strand. All right, that means this would be the 5' end of the daughter strand. That's 3' end. That's the direction it builds. Okay, here, however, is our lagging strand, and that's what we're going to focus on, right? So down here we have our first image of the lagging strand. So what we'll have is a primer placed right there. That's the primer that starts the first fragment, okay? All right, and there it is. So we have primase building our first fragment. That's going to give polymerase that critical 3' end it needs. And polymerase then, once that primer has set down, polymerase sits down and builds in or fills in this gap with daughter DNA until it hits the 5' end of the primer that actually starts the leading strand on the other half of the replication bubble, like I showed you. All right, while polymerase is building that fragment, primase here has actually moved further back, okay? The replication fork is over here and it's moving that way. Primase has leapfrogged backward toward it, and up here added another primer with a new 3' end. So polymerase, when it hits this primer, after it's built that fragment, it comes off. It's going to go backwards till it finds that new 3' end on that primer, and it fills in the next gap until it hits the 5' end of the primer on the fragment it built just before that. All right, once it hits that 5' end on that primer, it comes off. It's going to go back, and while it was doing this fragment, this Okazaki fragment, primase was back up here building yet another primer, right, again toward the fork, which is moving that way. Polymerase, once it comes off of this one, goes back, finds that next primer, fills in that gap. All right. So what's happening here is that polymerase is building each fragment in the 5 to 3 direction, but by leapfrogging backwards and finding the next primer, it is moving overall toward the replication fork, with the replication fork, right? So let me zoom in a little bit here. Oops, sorry. Okay. All right. So you can imagine that polymerase has just built this fragment, comes off, hits that primer, comes off, goes backwards, builds this fragment, hits the 5' end of that primer, comes off, goes backwards, hits this primer, fills in that gap, comes off. And so you can see how it's filling gaps, building fragments in the 5 to 3 direction, but leapfrogging backwards, which keeps it in line or in sync with the replication fork, which is moving that way. That is called lagging strand replication, all right? Now, we've got all these primers, right? And primers are made of RNA, not DNA. So that's a problem. That RNA has to be replaced with DNA. So the workhorse here is DNA polymerase 3. That's the one building the daughter DNA, all right? Once it's done, a different DNA polymerase, polymerase 1, comes in, plucks out the RNA nucleotides and replaces them with DNA nucleotides. Now, problem is that polymerase 1, when it gets to this last primer nucleotide and it pulls it out and replaces it with a DNA nucleotide, it cannot seal that last gap in the sugar phosphate backbone. So what happens is that we have DNA ligase, as I said before, that will come in and seal up all those gaps. There will be one gap where every primer was in that sugar phosphate backbone, and ligase will come in and seal up that gap once the polymerase 1 has moved out of the way. So what you end up with then on the lagging strand is a seamless, continuous coding strand of daughter DNA, same you have on the leading strand, but it was just built in a much more complicated way because of this anti-parallel nature of the DNA molecule. That's what I meant by the structure of the molecule dictating that it be copied in a very specific way, and there you see exactly that. All right, let's look at this image, which is showing you sort of multiple fragments all being built at the same time, right? So here, let's start on the right-hand side. Here we have ligase sealing the gap for where that first primer that was laid down was, right? Here we have polymerase removing the next primer, all right? Here we have our last primer that's in place. Beyond that, we have polymerase 3 still in process building a fragment. Down here we have primase putting down the next primer for polymerase to attach to and then build this fragment. So if you go from step 1 all the way up to step 5 here, you can see the process in sequence. But because we're moving in sequence this way, overall we're moving toward that replication fork, which keeps polymerase in line with it. And again, here you see leading strand. There was one primer up there. Once polymerase 3 sits down, it just goes and keeps up with the primer. That's leading strand replication. Very, very simple compared to lagging strand. All right, so let's sort of summarize again or rehash these different enzymes that are involved here. So helicase, like I said, is the enzyme that unwinds the DNA helix into the ladder conformation and separates the two parent strands from each other. We have single-stranded binding proteins then that sort of tack on to those separated strands, lock them down so they can't rejoin and coil back up, all right? We have topoisomerase that corrects the overwinding problem between these two forks as they grow together, right? As I said, prevents that knot from forming, which would prevent DNA replication. We have primase that puts down our RNA primer, giving polymerase 3 that critical 3' end that it needs, that free 3' end, all right? We have DNA polymerase 3, which is sort of the workhorse doing all the building, right? Adding daughter DNA nucleotides to build that daughter strand onto the template parent strand. And again, always building it in the 5 to 3 direction, like I said. Polymerase 1 coming in, removing the RNA primers, replacing them with DNA nucleotides, and ligase sealing the gap where each primer used to be, joining all the Okazaki fragments together into one continuous strand of DNA. All right, so next I would have shown you this BioFlix animation, but again, it's not working on my iPad for some reason. But again, it's also on Blackboard. You can go there, you'll see this image of it, you can watch it, and it explains all this pretty well, okay? All right. Next thing to talk about is the proofreading mechanism that I mentioned before. I said DNA replication is very accurate, okay? Now, that process we just went through is very complicated, and so those DNA polymerase 3 enzymes, they make more mistakes than you would think. But most of those mistakes are caught and corrected after polymerase moves on, okay? First thing we have is basically what's called mismatch repair, right? Write this down. So, mismatch DNA repair. So in this case, repair enzymes will correct errors in base pairing. So let's say, for example, we have an A on the parent strand, then over here you would think, well, we should have a T, right? But instead we get a C. That is a mismatch. All right, that's a mutation that needs to be fixed. So once DNA polymerase 3 moves on, after having made that mistake, repair enzymes will come in, identify that mistake, pull out that cytosine nucleotide, and polymerase will then come back in and correct it by adding a thymine nucleotide. Remember, A with T, right? Okay. Now, what kinds of things can cause mismatches like this? Well, you think about it. Your DNA is being copied all the time. Every second of your life, you have new cells being produced through mitosis. And what happens during S phase before the mitotic phase? DNA is copied. So when you're laying on the beach or you're out working in the sun and you don't have sunscreen on, your skin cells are being copied. And their DNA is being replicated in preparation for that. So while your DNA is being replicated, it's being bombarded by UVA and UVB radiation, and that can cause DNA polymerase to make more mistakes than it normally would. Those are mutations. If they're not caught, they could lead to a skin cancer situation, right? And that's the basis of skin cancer, is that you have DNA polymerase copying DNA while your cells are being bombarded by UV radiation because you're not wearing sunscreen, let's say, or you have exposed skin exposed to sunlight. And that's what is the basis of skin cancer, okay? So DNA can be damaged by exposure to harmful chemical or physical agents, even such as cigarette smoke. X-rays can do that, UVA, UVB, as I said. A lot of industrial solvents that we find as pollutants in the environment, that sort of thing. There are all kinds of agents that can cause mutations in our DNA. Those agents are called mutagens. So a mutagen is just any chemical or physical agent. Physical agent, UV rays, X-rays, chemical agent, solvents, pollutants, cigarette smoke, chemicals, those kinds of things, all right? Now, in some cases, you have an error that's much more complicated than a simple mismatch in a single base pair. You can have several base pairs that have an error, and in this case, what you need is an enzyme called a nuclease, all right, a nuclease. This carries out what's called nucleotide excision repair. So in the case of nucleotide excision repair, an entire stretch of damaged DNA can be lifted out and corrected. That's what you see here. So take a look at these two bases in this strand of the parent DNA. They're actually bonded to each other, covalently bonded to each other right there. That is called a thymine dimer. Okay, so these are two thymine nucleotides, and rather than hydrogen bonding to the adenines on the other side, they have covalently bonded to each other. It's thought that UV radiation causes those, right? So those are a problem. What will happen then is that if that problem is recognized, is a nuclease enzyme will come in, make cuts on either side of that, lift out that damaged section. DNA polymerase comes in and fills in that gap there, with the correct DNA sequence, right? And then again, because it can never fill in that last break in the sugar phosphate backbone, ligase comes in, seals the gap. So that is DNA excision repair, okay, or nucleotide excision repair. All right. So what is the evolutionary significance of these errors that occur in our DNA as a result of mutations or DNA replication mistakes? Well, the error rate after proofreading is low, but it's not zero. Some of these mistakes can sort of fall through the cracks. And if they are occurring in cells that become sperm or egg, they can be passed on as mutations to the next generation. Those mutations are what causes different versions of genes to form. Remember we talked about in the last chapter on Mendelian genetics, alleles being new versions or different versions of the same gene. The only way you get alleles is through mutations, right? And mutations are errors that occur during DNA replication. So these errors that occur that are not corrected and fall through the cracks, they can produce new versions of genes, alleles, and that's the basis of all the genetic diversity we see around us. It's why no two humans, with the exception of identical twins, which is a different process, are exactly the same. It's why we're all different people. It's why we have different species. This genetic variation that results from past mutation is the raw material for evolution by natural selection to operate on. If mutation didn't occur, then you wouldn't have different species. We might have the original bacterium that, began life on this planet 3.8 billion years ago, and that might be it. So this process then of mutation is very important. It allows evolution to operate, essentially. Okay. All right. So that's the end then of chapter 16. All right.